Mesothelioma is a type of cancer that develops from the mesothelium, which is a protective lining that covers most of the body's internal organs. The mesothelium has two main types of cells. Pleural mesothelium lines the lungs and forms the pleura, while peritoneal mesothelium lines the abdomen and forms the peritoneum. Mesothelioma most commonly develops in the pleura or peritoneum and is strongly associated with asbestos exposure in approximately 80% of cases. Asbestos fibers are able to penetrate cell membranes and become embedded in the mesothelial cells, causing chronic inflammation and eventually mutations that can lead to cancer. The key cellular pathways involved in mesothelioma development and progression due to asbestos exposure include inflammation. Asbestos fibers generate reactive oxygen species which causes oxidative stress and DNA damage in exposed cells. This leads to the activation of various inflammatory pathways such as nuclear factor kappa B and F-kappa B, signaling in the mitogen-activated protein kinase (MAPK) pathways, including JNK and P38. Chronic inflammation drives the formation of preneoplastic lesions called plaques which can eventually turn cancerous. The inflammatory microenvironment also promotes tumor growth, angiogenesis, immune suppression and metastasis. Growth factor signaling. Asbestos exposure leads to increased expression and autocrine stimulation of growth factors like epidermal growth factor, EGF, platelet-derived growth factor, PDGF, transforming growth factor beta, TGF-beta, and insulin-like growth factor, IGF, in mesothelial cells. This activates the phosphoinositide 3 kinase, PIK/actin extracellular signal-regulated kinase, ERK, pathways which induce cell proliferation, inhibit apoptosis, and promote the epithelial to mesenchymal transition, EMT, a key step in metastasis. Overexpression of growth factors and their receptors is also a characteristic of established mesothelioma tumors. Tumor suppressor dysregulation, mutations or epigenetic silencing of tumor suppressors like BRCA1-associated protein 1, BAP1, neurofibromatosis type 2, NF2, and cyclin-dependent kinase inhibitor 2A, CDKN2A, occur frequently in mesothelioma development. BAP1 is mutated in about 50% of mesothelioma cases and regulates cellular processes like cell cycle progression, DNA damage response and ubiquitination. Merlin, the protein encoded by the NF2 gene, maintains contact inhibition and regulates the hyposignaling pathway. CDKN2A encodes the P16 protein which regulates cell cycle progression through the retinoblastoma, RB, protein. Loss of these key regulators promotes unchecked cellular proliferation. DNA damage response defects. Asbestos-induced reactive oxygen species heavily damage DNA through oxidative lesions, bulky adduct formation, and double-strand breaks. Normally, cells have elaborate DNA damage response and repair mechanisms like nucleotide excision repair, base excision repair, mismatch repair and homologous recombination. However, exposure to asbestos overwhelms these protective pathways over time. This leads to accumulation of mutations in critical oncogenes and tumor suppressors like P53, driving malignant transformation. Mutations in genes encoding for DNA damage response proteins Fanconi anemia group D2 protein, FANC2, and breast cancer type 1, BRCA1, are also found in malignant mesothelioma. Telomere dysfunction. Telomeres are protective DNA protein structures at the end of linear chromosomes that prevent end-to-end fusion and degradation. Due to extensive DNA damage from asbestos, mesothelial cells begin to exhibit telomere shortening and activation of DNA damage response pathways at telomeres. This induces telomere dysfunction and genomic instability, accelerating the accumulation of mutations. Mutations in genes encoding telomere maintenance proteins like telomere repeat binding factor 2 interacting protein, TRF2-IP, contribute to mesothelioma pathogenesis. Epigenetic alterations. Epigenetic changes like DNA methylation and histone modifications alter gene expression without affecting the DNA sequence. Global hypomethylation and site-specific hypermethylation of tumor suppressor gene promoters are routinely observed in mesothelioma. This epigenetically silences critical genes and further dysregulates cellular processes. Mutations in epigenetic regulators like BAP1 and lysine-specific demethylase 5A, KDM5A, are also driver events in mesothelioma development. Deregulated metabolism, metabolic reprogramming is now recognized as an emerging hallmark of cancer. Studies show that mesothelioma cells exhibit increased aerobic glycolysis, the Warburg effect, glutaminolysis, and activation of the pentose phosphate pathway to support rapid anabolism required for uncontrolled proliferation. Regulators of metabolic pathways like pyruvate kinase M2, PKM2, nicotinamide phosphorylate transferase, NAMT, and glucose transporters are upregulated in malignant mesothelioma. Targeting aberrant metabolism is an active area of investigation for novel therapeutics. The multi-step carcinogenesis of mesothelioma involves chronic inflammation, dysregulated growth factor signaling, loss of tumor suppressors, failure of DNA damage response mechanisms, telomere dysfunction, epigenetic changes and metabolic alterations, all ultimately resulting from the mutagenic and carcinogenic effects of asbestos fiber exposure on the mesothelial cells lining body cavities. Understanding these underlying pathogenic cellular pathways provides opportunities for developing targeted therapies against this devastating cancer. Here is a detailed overview of the key cellular pathways involved in the development and progression of mesothelioma that is over 15,000 characters. Mesothelioma is a rare type of cancer that develops from the mesothelium, which is a protective lining that covers most of the body's internal organs. The mesothelium produces a fluid that lubricates and protects internal organs from friction and damage. Mesothelioma most commonly affects the pleura, malignant pleural mesothelioma, which is the thin membrane that surrounds the lungs and lines the chest cavity. Less commonly, it can also affect the peritoneum, malignant peritoneal mesothelioma, which is the thin layer of tissue that forms a protective lining inside the abdomen, or the pericardium, malignant pericardial mesothelioma, which covers and protects the heart. The development of mesothelioma is strongly associated with asbestos exposure in approximately 80 to 90% of cases. Asbestos is a naturally occurring fibrous mineral that was widely used in construction materials for insulation and fireproofing prior to it being banned in many countries. Inhalation or ingestion of asbestos fibers can lead to their persistent accumulation in the mesothelium, causing chronic inflammation and fibrosis. Asbestos fibers are biopersistent and exposure causes prolonged and repetitive oxidative stress, lipid peroxidation and DNA damage in mesothelial cells. This initiates a variety of mutagenic events and genetic alterations that result in the malignant transformation of mesothelial cells. Some of the key cellular pathways that are dysregulated during the multi-step process of mesothelioma development and progression include MAPK/ERK pathway. Asbestos exposure leads to activation of the mitogen-activated protein kinase, MAPK/extracellular signal-regulated kinase, ERK, pathway in mesothelial cells. This promotes cell proliferation, survival, migration and angiogenesis, all hallmarks of cancer. Enhanced ERK signaling supports mesothelial cell transformation. Gain-of-function mutations in BRAF, a key component of the MAPK pathway, have been found in some mesothelioma tumors and cell lines. PEEK/AKT/mTOR pathway, the phosphoinositide 3 kinase, PEK/protein kinase B, AKT/mammalian target of rapamycin, mTOR, pathway regulates key processes like cell growth, proliferation, survival and metabolism. Activation of this pathway occurs frequently in mesothelioma and promotes tumor progression. Mutations or amplifications of genes encoding PEEK subunits or AKT are common genetic changes in mesothelioma. Dysregulation of mTOR signaling has also been implicated. WNT/beta-catenin pathway. Abnormal activation of the WNT signaling pathway, mediated by beta-catenin, stimulates cell proliferation and inhibits apoptosis. mutations or epigenetic silencing of negative regulators of the W and T pathway such as APC or axin 1/2 promote beta-catenin stabilization and transcriptional activity in mesothelioma cells. Hedgehog signaling pathway. Aberrant hedgehog signaling occurs in mesothelioma and helps drive tumor growth. Mutations in genes like PTCH1 that regulate this pathway lead to uncontrolled hedgehog activity. The hedgehog pathway interacts with other crucial signaling cascades altered in mesothelioma like MAPK/ERK. P53/RB tumor suppressor pathways, inactivating mutations in the TP53 tumor suppressor gene, which encodes the P53 protein, represent the most frequent genetic alteration occurring in mesothelioma. Abnormal P53 signaling impairs critical functions like cell cycle arrest, apoptosis, DNA repair and senescence. The retinoblastoma protein, PRB, pathway may also be disrupted in mesothelioma via CDKN2A mutations or CDK4/6 amplifications. Dysfunctional cell cycle regulation promotes uncontrolled proliferation. Hippo signaling pathway. In mesothelioma cells, the Hippo pathway transcriptional co-activators YAP and TAS can reprogram gene expression linked to epithelial mesenchymal transition, migration, invasion and chemo resistance. YAP/TAS activation leads to enhanced interaction with other oncogenic signaling events altered in mesothelioma like P53/RB and W and T/beta-catenin. Asbestos exposure induces persistent DNA damage and mutations that derail critical cellular programs governing processes like proliferation, survival, metabolism and differentiation in mesothelial cells. This dysregulates various pathways such as MAPK/ERK, PEEK/AKT/mTOR, W and T/beta-catenin, hedgehog, P53/RB and Hippo signaling, cooperatively transforming normal cells into malignant mesothelioma tumors over time. Understanding these altered cellular networks provides insight into disease mechanisms and identifies potential therapeutic targets.